Hello everybody. Welcome to ITLS Academy. Today I'm going to start with the topic bioseparation process. So first of all, what is bioseparation? If we talk about the bioseparation, it refers to the systematic study of the scientific and engineering principles utilized for the large scale purification of biological products. Basically, it is used to purify the biological products on a large scale. The main goal of bioseparation is to refine molecules, cells, and parts of cells into purified fractions. So, while discussing about the bioseparation, we should also know what is bioprocessing. Basically, bioprocess is a specific process that uses complete living cells or their components to obtain desired products. This has been divided into two categories. The first one is reactive bioprocessing and the second one is extractive bioprocessing. In reactive bioprocessing, the bioseparation process follows some form of biological reactions. In extractive bioprocessing, almost entirely involves bioseparation. In reactive bioprocessing, the upstream processing involves steps such as biocatalyst screening, enrichment, isolation and propagation, cell manipulation by recombinant DNA technology, media optimization and formulation. The biological reaction involved could be fermentation, cell culture, or simply an enzymatic reaction. While in extractive bioseparation, upstream processing involves raw material acquisition and pretreatment. So next, here are the both categories which have been explained with the help of a flowchart. In both the categories, upstream process takes place. So in reactive bioprocessing, first we should start with the reactive bioprocessing. In this reactive bioprocessing, the first step which is taking place is upstream process. So we should also know what is upstream process. Basically, it is the identification and extraction of raw materials. It is the initial process of fermentation, and we all know that fermentation, it's a boiling process. It is a chemical conversion of organic substances into simpler compounds. So the first step which is taking place is upstream process, and after the upstream process the biological reaction takes place and the final bioseparation, and we are getting the finalized biological product. The next, in extractive bioprocess, again the upstream processing taking place and final the bioseparation, and we are getting the finalized biological product. So next, what is being separated in bioseparation? So if we talk about the separation, then the biological products and biological products are being separated and we are getting the finalized purified form. So biologically derived products can be categorized in different ways. One way being based on their chemical nature, which has been shown in table first, and the next, biological products can also be classified based on their intent applications, which has been shown in table second. So here is the table first, in which biological products are being classified based on their chemical nature. The first, here are the categories with examples. So the first one is solvent. The examples are ethanol, acetone, butanol. The next category is organic acid. Examples are citric acid, lactic acid, butyric acid. The next is vitamin. Examples are ascorbic acid, vitamin B12. The next is amino acids. Examples are lysine, phenylalanine, glycine. The next is antibiotics. Examples are penicillin, streptomycin, gentamicin. The next category is sugar and carbohydrates. The example are glucose, fructose, starch, dextran. And the next is lipids. Examples are glycerol, fatty acids, corticosteroids. The next category is nucleic acids. The examples are plasmid, therapeutic DNA, retroviral vectors, antisense oligonucleotides, ribozymes. The next category is semi-purified proteins. The examples are industrial enzymes, egg proteins, milk proteins, whey proteins, soya proteins. The next is purified proteins. Examples are therapeutic enzymes, monoclonal antibodies, plasma proteins, cytokinins, interleukins, hormones, growth factors, diagnostic enzymes, vaccines. The next is cells. The examples are Baker's yeast, Bewer's yeast, freeze-dried lactobacillus. Then the second last category is crude cellular extract. The example are yeast extract, soya extracts, animal tissue extract, and the last category is hydrolysates. The examples are soya hydrolysates, whey hydrolysates, animal tissue hydrolysates. Next is the table second, where the biological products are being classified based on their application. So here are the categories with some examples. The first category is industrial chemicals. The examples are solvents, organic acids, industrial enzymes. The next category is agrochemicals. The examples are biofertilizers, biopesticides. The next category is biopharmaceuticals. The examples are antibiotics, hormones, monoclonal antibodies, plasma proteins, vaccines, hormones, cytokinins, and therapeutic nucleic acids. The next category is food and food additives. Examples are whey proteins, milk proteins, egg proteins, soya proteins, protein hydrolysates. The next category is nutraceuticals. Examples are vitamins, enzymes, coenzymes, cofactors, amino acids, purified whey proteins. The next category is diagnostic products. The examples are glucose oxidase, peroxidase. The next category is commodity chemicals. The examples are detergent enzymes, insecticides. And the second last category is laboratory reagents. The examples are bovine serum albumin, albumin, lysozyme. The last category is cosmetic products. The examples are plant extracts and animal tissue extracts. So next, for a good bioseparation process, one should ensure desired purity of the product. It also ensures the stability of product. It keeps the low cost, and it is reproducible, and it is scalable, and at last it meets the regulatory guidelines. So here it was all about the brief intro of bioseparation process. So in the next lecture we will study about the basis of separation of bioseparation process. So follow ITLS Academy on Instagram, Facebook, Twitter. Register at www.itlsacademy.com, and for any query we have a helpline number, that is 7080833450. Thank you for being with us.